Dear friends and my dear students, I am very sorry for a long gap of about 15 days. It was because of my engagement somewhere else. Now again we are resuming the class of biochemistry. In the previous sessions, I had discussed amino acids, proteins and enzymes. Today I am discussing lipids. Lipid is one of the most important constituent of our body and also a source of energy. But the term lipid is a vague term. It has many unrelated compounds, and the name lipid does not signify any specific chemical group. At first, we will discuss about the fatty acids and the glycerides. Lipid is a highly diversified group of organic compounds which are insoluble in water but soluble in organic solvents like alcohol, ether, benzene, acetone, chloroform, hexane, etc. So the definition is based on a physical property, that is, its solubility in a specific media. Therefore, the physical property of various compounds may be similar, and therefore the lipid does not make one single group of chemicals, but it is varying chemical nature. You can classify lipid in at least three groups. One is the fatty acid derivatives. When it forms esters with glycerol, these are called glycerides. The neutral fats and oils, acidic fats and oils are glycerides. The fatty acids can also form ester with monohydric alcohol, such as in case of wax and shellac. Quite a different group is the esterols and steroids, that is the compounds derived from the esterol, and the third group includes some odd compounds, mostly of plant origin. So at first we have to begin with fatty acids and their derivatives. Now let us discuss what is a fatty acid. The higher monobasic acids present in fats and oils are called fatty acids. Practically, the compound with 12 or more carbon atoms are called fatty acids, as these are generally non-volatile at normal room temperature. Up to 6 carbon atoms, people say them simply organic acids, but 7 to 11 carbon has an intermediate position. They become volatile at the higher temperature, that is above the 25 degree. But those compounds having 12 or more carbon are normally non-volatile at the room temperature, that is 25 degree. They may be saturated or unsaturated compounds and may contain odd or even number of carbon atoms. The natural fatty acids have generally even number of carbon atoms. Odd carbon compounds have odd behavior, not only in their chemical properties but also in physical properties. The organic compounds have some interesting properties which affects the melting and boiling points of the fatty acids also. We are, however, mainly concerned with melting point only because the boiling point is not achieved in our body. These properties are: the melting and boiling points of all organic compounds increase with increase in their molecular weight. That is, practically the melting and boiling point of 10 carbon compound is less than 14 carbon compounds, and 14 carbon compounds have melting point and boiling points less than 16 carbon compounds, and so on. Similarly, the odd carbon compounds, the 13 carbon compounds, have melting point lower than 15 carbon compounds, and those of 15 carbon compounds is lower than 17 carbon compound. The melting and boiling points decrease in unsaturated compounds, and these are directly affected by the number of double bonds, though the molecular weight decreases very less, only 2 per double bond. Therefore, melting point may become lower than many of the smaller compounds. The melting and boiling points of odd carbon compounds is lower than even its predecessor, even carbon compounds. In this graph, you can see that the melting point of saturated drawn in upper line and unsaturated drawn in lower line. The same lines are used for the even carbon compound and odd carbon compound also. The even carbon compounds, say 12, 14, 16, 18, 20, 22, 24, etc., have a regular pattern in the upper line. Similarly, the odd carbon compound 11, 13, 15, 17, 19, 21, 23, etc. have lower line. That is, the melting point and boiling point of the odd carbon compound is less than the even carbon compound. Similarly, the saturated compounds have higher melting point and the unsaturated compounds have lower melting points. Let us see by some examples what are the saturated carbon compounds and unsaturated carbon compound. Here is palmitoleic acid made of 16 carbon with one unsaturated bond in between 9 and 10 carbon atoms. Another is oleic acid of 18 carbon. It also has a double bond between 9th and 10th carbon atoms. The third is vaccinic acid made of 18 carbon, but the double bond is in between 11th and 12th carbon. The fourth compound is linoleic acid with two unsaturated bonds, one between 9 and 10 and another between 12 and 13 carbon atoms. Another compound with 3 unsaturated bonds have double bonds in between 9, 10, 12, 13 and 15, 16 carbon atoms. So these unsaturated compounds have lower melting point than their saturated relatives. The melting point decreases from one double bond to two double bonds and further in case of three double bonds. That is, oleic acid and vaccinic acid having a single double bond have almost similar melting points, but linoleic acid has lower melting point and linolenic acid has very low melting points. Arachidonic acid found in Arachis hypogaea, that is groundnut, have 12 carbon, but the unsaturated bonds are between 5th, 6th, 8th, 9th, 11th, 12th and 14th, 15th carbon atoms. Because of the double bonds at 4 places, arachidonic acid also has lower melting point. Now, let us have a list of some common fatty acids. The saturated fatty acids are lauric acid with 12 carbon, found in spermaceti, cinnamon, palm kernel, coconut oil, laurels and butter. Myristic acid having 14 carbon is found in nutmeg, palm kernel, coconut oil, myrtles and butter. Palmitic acid with 16 carbon is common, though the name has been given on the palm oil. Stearic acid, it is another common fatty acid with 18 carbon. It is common in the stearin derived from the milk cream. Arachidic acid is saturated, found in peanut and groundnut, and its unsaturated relative was arachidonic acid. This is the list of some unsaturated fatty acids. Palmitoleic acid with one unsaturated bond, most common in fats and oil. Oleic acid, it also has one unsaturated bond, but at different place. It is again common in the fats and oil. Vaccinic acid has 18 carbon and unsaturated bond between 11th and 12th carbon. It is the third common fatty acid in fats and oil. Linoleic acid had two unsaturated bonds found in peanut, cotton, seed and soybean. Linolenic acid made of 18 carbon has three unsaturated bonds. It is rare, found in the seeds of evening primrose and linseed. Arachidonic acid, that I earlier said, it is found in groundnut. Now see the structure of arachidonic acid. You can draw the line for the single and double bonds. There are 4 double bonds and 20 carbon atoms. The double bonds are between 5th, 6th, 8th, 9th, 11th, 12th, 14th, 15th carbon atoms. This is another diagram for the same, showing 4 unsaturated bonds. Now in natural fats and oils, even carbon compounds 12 carbon, 14 carbon, 16 carbon or 18 carbon etc. are more common. The 16 carbon compound is most common, but the odd carbon compounds are rare. Likewise, the unsaturated forms of 16 carbon and 18 carbon compounds are more common, whereas in the oil, 14 carbon and 12 carbon compounds also are found to decrease the melting points. Now for the human and other animals, there are few essential fatty acids. The animals have no enzyme to put a double bond ahead ninth carbon. Therefore, they have to take linoleic acid, linolenic acid, and arachidonic acid from plants or other sources in food. These are essential constituents of animal fats, so these are known as essential fatty acids. Arachidonic acid is conditional essential fatty acid. It is not needed in all metabolism. Now, what is the triglyceride? Glycerol is a trihydric alcohol, that is, all the three carbon atoms of glycerol contain a hydroxyl or alcohol group, so it can form esters with fatty acids at all the three positions C1, C2 and C3. Accordingly, it can form monoglyceride, diglyceride and triglyceride. That is the name of bond between glycerol and fatty acid. The ester bond between glycerol and fatty acid is called glyceride. In the triglycerides, when all the three fatty acids are saturated, the melting point is very high, above the body temperature. So such esters are not formed in animals and plants. In most animal fats, one fatty acid is unsaturated, so the melting point decreases to 35-36 degree. It can be stored well and mobilized when there is an increase in metabolic rate of body. When there are 2 or 3 unsaturated fatty acids, the melting point further decreases and the triglycerides remain in liquid state at room temperature. These are called vials. It is therefore not stored in animal body, in which body temperature tends to remain higher at 37 degrees centigrade. The plants that store such esters in their seeds therefore grow in winter. See in this diagram, there are 3 fatty acids with alkyl group R, R dash, R double dash. They form ester with glycerol and form the triglyceride. See a table to denote how the melting point decreases. If there are 3 palmitic acid in the triglyceride, the melting point is 66 degree. When 2 palmitic acid and 1 palmitoleic acid is there, the melting point decreases to 36 degree. So it is best stored in mammalian body that had normal temperature 37 degree. When there is a single palmitic acid but 2 palmitoleic acids, the melting point decreased to 14 degrees Celsius. So it is an vial. And when all the 3 fatty acids are palmitoleic acid, that is unsaturated fatty acid, the melting point decreases to become below 0. Now, when on the 3rd carbon, in place of a fatty acid, a phosphate is attached, it forms the phospholipid. Phospholipids, phosphoric acid is tribasic acid, but only one active hydrogen atom is utilized in ester bond formation. Its 2 active hydrogen atoms remain unreacted, and therefore the compound acts as a strong acid. It therefore forms acidic fat and oil. In more than 90% cases, A saturated fatty acid is found at first carbon, unsaturated fatty acid at second carbon, and they remain in liquid state inside the body. However, the phosphate of phospholipid may further react with other molecules of glycerol, alcohol groups of other compounds, and amine, etc. In this way, a phosphatidyl compound is formed. Phospholipid plus glycerol, it is phosphatidyl glycerol. When two phospholipids attach to one glycerol, diphosphatidyl glycerol is formed. Phospholipid plus inositol is phosphatidyl inositol. Phospholipid plus serine is phosphatidyl serine. When ethanolamine becomes attached, it is called phosphatidyl ethanolamine. When choline is attached, it is phosphatidyl choline, and so on. Phosphatidyl choline is commonly known as lecithin, which forms major part of egg yolk, whereas phosphatidyl ethanolamine is commonly known as cephalin, that is found in the myelin sheath of nervous system. Sometimes at carbon 1, a fatty aldehyde, that is a long chain aldehyde, is attached by ether bond. Then the compound is called phosphatidyl compound instead of phosphatidyl compound. Here L is for the aldehyde. In wax and shellac, a long chain fatty acid becomes esterified with a long chain alcohol. These are esters of fatty acids such as myrtic acid with higher monohydric alcohol such as acetyl alcohol. They have very high melting points. These are produced by the insects. Wax is secreted by honeybee of genus Apis, and shellac is secreted by lac insect Laxifer lacca and used in building of their combs. These compounds cannot be digested or metabolized in our body or any other animal's body. Now come to a very mysterious compound known as sterol and the steroids. The steroids form a biologically very important group which are synthesized mainly in the animal's body. The plant steroids are rare. These are made of a nucleus named as perhydrocyclopentanophenoethrin containing total 17 carbon. Cholesterol is the mother compound which is synthesized mainly in liver, and it is made of 27 carbon. From this cholesterol, a number of other compounds can be synthesized. Various other forms can be synthesized in testes, ovaries, adrenal cortex. These are parts of animal fats including ghee. In liver, cholesterol converts into cholic acid, from which bile salts namely glycocolate and taurocolate are formed. They act as detergents and help in emulsification and dissolution of dietary fats in duodenum and ileum. A major amount of cholesterol passes into circulation. In case if the level of cholesterol is high, it is called hypercholesterolemia. It can be deposited in the wall of blood vessels, causing their thickening and resulting into high blood pressure. Deposition of bile salt in gallbladder causes formation of biliary stones or gallstones. This is the structure of cholesterol. You can see there are 27 carbon atoms. From cholesterol, the three hormones cortisol, corticosterone, aldosterone, these are found in adrenal cortex. Progesterone, estradiol are female sex hormones and testosterone is the male sex hormone. So as hormones, these six compounds are very important and they regulate our body metabolism as well as the sexual maturity, spermatogenesis and fertilization. In testes, the male hormone androgens are formed. Testosterone is the most potent male hormone. In ovaries, estrogen and progesterone are formed. Progesterone is formed from placenta also. In adrenal cortex, glucocorticoids, cortisone and cortisol, and the mineralocorticoid aldosterone is formed. In plants, there are many lipids produced as secondary metabolites or the byproduct of its metabolism. The terpenes and terpenoids are most important among them. Camphor is an important example of terpene. Abscisic acid, a plant hormone, is terpenoid. See the structure of camphor. It is a terpene and abscisic acid is terpenoid, that is derived from terpene. In the next session of biochemistry, we shall discuss nucleic acid. Thank you very much.